Good day everyone, welcome to lesson number 4 on plant coordination. This video, I decided to put in a wee bit in the beginning because then you can skip through wee bits if you don't want. So in this we're going to do a wee homework, summary, a review of that, look at the specification and then move on to new work, which is the last wee bit. This is a very short wee topic. So we've learned about phytochromes, and in this wee bit we learn about 3 more hormones and their effect upon plants. Okay, thank you for those who did their homework. For those who didn't get it in, listen, this is A2 stuff. It's highly likely this stuff will not be retaught next year. If we have another COVID year, we could be in issues. So you need to keep on top of this. Let's do the short day plants. Now the key thing about short day plants is, you need to get the word and the explanation, into your word about P730 inhibiting. So in this SDP, short day plant, in the second scenario, we get flowering. Most people got that. Why? Refer to the darkness. Darkness greater than critical. So what? Well, we get a P730 converted to P660 due to the darkness. Remember that, slow in the darkness. Therefore, inhibitory P730 removed. Those are the key bits. Say what happens, refer to the darkness, mention any conversion. In long day plants, now the key issue here is anywhere you go to say P730, try to get the idea that it stimulates flowering. So it's stimulatory. In the scenario here, number 1, okay, what we have is we get flowering. Why? Well, the darkness is below, less than the critical. So P730 accumulates and stimulates flowering. In the second one, we get no flowering. Why? Period of darkness is greater than the critical. So what? We get the slow conversion of 730 converted to P660 slowly in the darkness. So not enough stimulated P730 remains for flowering. I mean, you should notice those two effects should be different. Every one of them. The light effect should have a different effect on the short day, long day scenario or plant. The final one, it's got the same, well, not really over it. This may be actually only about 5 seconds. And even then you would say, yeah, we still got over the critical, but it's the continuous darkness has been broken. The flashlight rapidly converts P660 to P730. And so we get an accumulation of the stimulatory 730 and flowering is induced. So here we have the spec for this wee topic. But if you look, I mean, literally what I've tried to go through with you are some of the key phrases. Phytochromes are pigments, photoreceptive pigments. Where are they found? Their leaves. 2 interchangeable forms. What happens to 660? Under red light. What happens to 730? Under far red. Pardon me. And the whole idea that, it's not worded probably the easiest here, but the slow conversion at night, P730 converted back at night, that's all there. The idea that the night period is critical to remove 730, that flash, the rapid conversion during the day is very, very quick, but it's the night that really determines what goes on. And then short day plants, P730 needs to be removed. Long day plants, P730 needs to accumulate. So this is where we're saying short days, P730 active form inhibits. Here it stimulates. And then the last wee bit is that we can mess around with that photo period to get flowers out of season. So the final wee bit of this topic is about plant hormones, and the specification states very precisely the role of the plant hormones, or they refer to growth substances, in stem elongation. Now, some of this might be familiar because I'm going to show you now the 3 hormones. One of them you know of as auxin. Now, what I would love you to do is just pause the video, get a bit of scrap paper, scribble down everything you know about this hormone called auxin from GCSE. Okay, it was Unit 1 GCSE, so year 11. Give it a go. Okay, fair enough. Some of you could have linked auxin to a process called phototropism and about cells elongating. We're going to cover that in a lot of detail. 2 new hormones. The second one called cytokinin, and the third one called a funny old name called gibberellins. So the bottom line is you must rote learn there are 3 plant hormones. A, C, G, alphabetically I put them in: auxin, cytokinins, gibberellins. To summarise what they do. Now, basically this is GCSE. Auxins promote cell elongation in a part of the shoot just behind the tip. Okay, that's key for you to get there, that bit. Auxin is produced at the shoot tip. Now, so at a level they might call it apical, meaning the very top of a shoot. Meristem is an actually growing bit. And it's auxin that results in phototropism. Now, here's the issue. Some years ago, we had to study all the experiments about it. They say removed that from the spec, but this still remains. So you can still get an interpretation on how it works, even though it's not directly mentioned in the spec. So we teach it now anyway. The second hormone is called cytokinins. And cytokinins just speeds up mitosis, promotes cell division. So basically, if you get something with more cells, it tends to grow, get bigger. And gibberellin is a funny wee one. I'm going to show you a wee experiment that somebody did. Now, it mentions here internodes. If you picture, let's even pick the right colour. If you picture that being a stem, well, you'll know that as wee stalks come out and wee leaves come out, and from basically primary science, you know there's another wee leaf there. People tend to grow it. Internode, that's a node, and that's a node, the junction of a leaf stem or a wee bud can be in there as a node. The internode is the bit in between. And so if you look, this one very specifically has got a key term in it. So see, love asking you about that. If I went to draw a cabbage, which I probably can't draw, so let's pretend this is a cabbage or a lettuce. All the leaves, this is very short, so all the leaves of the plant are very tight together. And a very famous experiment was this one. So it's a nice old one because the man's in black and white and also he wears a tie. So this is what the plant used to look like. So the leaves are fairly tight together. If you put gibberellins on it, look at the bit between those leaves. There's a leaf and there's a leaf. That has been elongated. So the whole plant is shot up. So a tremendous bit of growth in the internodal regions. So just now, pause and learn. So from GCSE, this is what I'm sure plenty of you would have thought about. Now we have a chemical hormone called auxin. It's produced in the tip, so this bit up here is the tip. This is sort of your fingers, like your fingernail is, obviously painted, and where the tip of it is, the auxin is produced, and auxin diffuses down. Now they may use the word coleoptile. Coleoptile just means that we first bit of shoot out of the ground, because they did a lot of experiments on cereal crops like oat and wheat, and that wee bit with a rolled leaf, just as a green single shoot sticking up, they call coleoptile. So don't worry. Auxin produced at tip, 1 mark. Diffuses downwards, 2 marks. Now where it works is in a special part called the zone of elongation. So that's these cells just below the tip. And what, again, you should be able to tell me is that auxin moves away from the light, so it moves to the darkened side. So as it comes down, it moves across, causing the cells on the darkened or shaded side to elongate more. Now it's differential. These still elongate. So this cell here is slightly longer than this one. So it's differential elongation. The cells on dark side elongate, and the bit they say in there indeed is the word more. Okay, so I will give you the PDFs for this next wee bit that are down below. In fact, I'll give you the PDFs for it all. So let me just take you through this, and then just going to give you time to read it, and then I have a homework question on phytochromes for you. So it's all about auxins. The process is phototropism. Tropism means movement. Photo obviously means in response to light. So auxin produced at the tip. It moves away to the shaded side, so lateral movement. If light from one side, they might say unidirectional light. It diffuses down the shaded side, causes them to elongate more than the illuminated side, causing it to bend towards light. Now here's some wee bit of detail, this wee bit here. How does it do that? Well, we know that cell walls are made of cellulose. We know those are fibres that are all overlapping each other. And you'll see written in here, very small, auxin loosens the connections between the microfibrils. And this is where AA2 can get synoptic, obviously, because we know it is the cell wall that inhibits water entry. So if we loosen those, well, there's going to be a water potential gradient, water's going to enter, and the whole cell swells up due to water entry into the vacuole by osmosis. And it's that more of that happening on the dark side causes it to bend towards the light. Why would you want it to bend towards light? Well, they want you to say, obviously, just like GCSE, greater absorption of light, greater photosynthesis. Now, auxins can be sprayed on plants, just simply to make them grow better. If you give both sides of it, it will grow up. So that's a wee bit of extra detail. There's lots of experiments associated with this. You might see some crop up in questions, but you're not required to know the names of any of the workers, but you may be asked to interpret on the basis that auxin causes cells to elongate. If it goes to one side, they elongate more. So that's that first wee topic finished. Just 3 things on it. Obviously phytochromes, auxins, cytokinins and gibberellins. Now I want you to log that and do the questions that I will send. Thank you.